All right, so welcome again. Uh, as I mentioned, we'll describe the work of the three branches in the Influenza Emerging Infectious Disease Division this afternoon. The three branches are Vaccines Research and Development, Pandemic Vaccines and Adjuvants, and Therapeutics. I'm Jonathan Seals. I'm the Acting Chief of the Vaccines R&D Branch. I'll start off, and I'll be followed by Christine Oshansky and Kim Armstrong, who are the chiefs of the other two divisions, respectively. Our division's mission, preparation for a possible influenza pandemic, remains a high priority for BARDA, as it has been since our creation in 2006. Much has been accomplished since then, but we continue to seek ways to increase our level of preparedness and response capability by building on the progress that we've made in the past 15 years. So I think in our presentations, you'll hear a little bit about the directions we hope to go in all three branches, again, to increase our level of preparedness for an influenza pandemic. In the Vaccines Research and Development Branch, our goal has been to ensure that vaccines able to prevent disease caused by pandemic flu viruses have been developed, and that they can be deployed in a timely manner and in amounts sufficient to mitigate the public health impact of a quickly spreading outbreak. Through our programs, the capability to mount such a response has increased greatly since 2006. Oops, sorry. Yep. There you go. And key to the progress we've made has been the recognition that pandemic flu preparedness and response capability could most realistically be met by and be built on and sustained by commercial seasonal flu vaccine enterprise. The problem in 2006 was that this basic infrastructure was far from robust at that time. Companies had actually been leaving the flu vaccine business over the preceding years, and for the 2004-2005 flu season, there was a single company supplying seasonal flu vaccine to the U.S. So as we applied BARDA's public-private partnership model to the initial preparedness efforts, we worked with vaccine manufacturers to support improvements in both seasonal and pandemic infrastructure. 15 years later, the commercial seasonal flu vaccine market has been reinvigorated, both in terms of products and formulations available, but also in terms of manufacturing technologies and capacity in the U.S. Most importantly for our mission, prototype pandemic vaccine formulations had been identified and subjected to regulatory review so that a credible response to an outbreak could now be mounted via our partnerships with the manufacturers. So looking at the purple columns there and the darker area in the top, we estimate now that in response to a pandemic flu outbreak, we can have vaccine available within several months, depending on whether the virus is one we've worked with previously or whether it's completely novel. We think we can have enough doses to administer two to each person in the United States within about 7 months, and we can have everyone fully immunized in less than a year. So these are notable advancements over where we were 15 years ago. We also recognize that there's room for further improvement in this capability, particularly in terms of the speed of response and the effectiveness of the vaccines. So our overall strategy has always been built on utilizing the best current technologies available at that time in order to enable response promptly, since we never know when an outbreak might occur, but also to continue looking for improvement in our capabilities as better technologies emerge. So this is the position we were in back in 2019 to 2020. And as we prepared to move into this next stage of improving our capabilities, we experienced an actual pandemic, but not of the type we were anticipating with flu. Of course, it was the COVID outbreak. And so for the next almost 3 years, everyone in our branch and in our division has been working pretty exclusively on the COVID response, and our flu programs have been essentially on hiatus during that period. But now we're in the process of refocusing on flu, and our overall goals for faster, better vaccines remain as they were 3 years ago. But we now have a large body of real pandemic experience on which to capitalize as we refine our thinking about the future for pandemic flu vaccine preparedness. So one outcome of this has been to refine our response goals, not just for flu, but for any newly emerging virus as outlined in the American Pandemic Preparedness Plan. So if you look at the green column, The goals at the top are those listed in the AP3, and they call for first doses of vaccine against the new virus to be available within 100 days after the nucleic acid sequence of the virus is available, for sufficient doses to immunize the entire U.S. population, approximately 660 million doses, to be available within 130 days, and to meet enough doses to meet global demand within 200 days. So these are ambitious goals, but they have in turn helped us to define our current initiatives for the next level of flu vaccine preparedness. So we need and we see possibilities for achieving success in terms of faster vaccine development, manufacturing and delivery technologies, and vaccines that can elicit a faster protective response in the near future. And we look at these interests in the next few slides. So as the vaccines R&D branch moves to refocus on flu and start to rebuild our portfolio, we have 4 areas of interest that we're focusing on. The first of these is faster vaccine platforms, and this is clearly learning from the COVID response, where we realize that there are now technologies for which vaccines can be developed and manufactured that will enable us to move much more quickly than the platforms that we're currently using for flu vaccines. So our first priority here is now to implement some of those technologies with flu vaccines, just as they were for COVID. Second priority is that in addition to making the vaccines faster, we need to get them into people's arms faster. And so our second priority is to look at faster vaccine delivery. And here again, there was some learning from the COVID response. And prior to the pandemic, we had concerns about the ability to get a large number of doses into people's arms quickly, both in terms of the availability of supplies, needles and syringes for use in intramuscular injection, but also trained personnel to administer the vaccines, and of course, the ability to ship these vaccines around in appropriate storage conditions. With COVID, the administration work actually went perhaps better than expected, but it was still a very tremendous undertaking. And so we think there are better ways that we can implement to get vaccines delivered into people's arms that may eliminate some of the need for cold chain, for needles and syringes, and may even enable self-administration. So our second focus will be on faster vaccine delivery, alternative delivery methods. The third area of focus is on improved vaccine performance. And this is an area which could cover potentially a broad number of approaches and projects. The ultimate goal, of course, is a universal flu vaccine that would allow us to address pandemic preparedness proactively. We don't see that as necessarily a short-term goal, but we think there are incremental improvements in vaccine efficacy which would allow us to have improved response capabilities. And one of those would be simply to enable vaccine formulations that would elicit protection with a single dose rather than 2 doses. Going from 2 doses to 1 would require then less vaccine and would of course require less time for people to achieve protection after that initial immunization. So that's our third area of focus. The fourth area applies to, of course, any new technology we developed. Just as we did in the first generation of influenza vaccine preparedness, it was key to establish capacity to produce the countermeasures that we had developed in sufficient quantities to supply the United States in an appropriate period of time. So as these new technologies come on board, we'll of course be looking to accomplish the same thing with those. So let's look a little more closely at the faster vaccine platform programs. So the obvious lesson from the COVID response is that RNA-based vaccine platforms have the potential to move much more quickly than the ones we are currently using for flu vaccines. And this will be our initial focus moving forward with flu vaccines for the next generation. Initially, I think we're going to bide our time as the technology is still evolving. The technologies used by Moderna and Pfizer were a first generation, but there are a number of other companies and organizations working on variations of RNA-based platforms. And I think our position is that we want to wait a little bit of time for that to work itself out before we move aggressively in the direction of implementing them for flu vaccines. So we anticipate that over the next year or two, But starting in fiscal year 24, I think our goal is to be able to make larger investments into moving mRNA-based platforms forward into advanced development and ultimately licensure for seasonal vaccines, and of course to demonstrate that they can be used for pandemic preparedness as well. Now the work that we're doing here, or proposing to do here with FASTR platforms, we're discussing this separately from the other areas of interest that I mentioned on the previous slide. But I should mention that we are also looking for synergies between all of these different areas of interest. It's certainly possible that FASTR platforms could also enable us to achieve better protection, for example, or faster protection. It's also possible that alternative delivery could be combined with some of these new platforms and achieve additional benefits as well. So we don't want to consider any of these initiatives in a vacuum, but potentially look for ways that they can work together. And then, of course, as I mentioned on in the initial slide, the American Pandemic Preparedness Plan does not refer just to flu, but to any new virus that emerges. And as we think about these new technologies in terms of platforms, they're potentially applicable not just to flu, but potentially to overall emerging infectious disease preparedness. So we want to keep all these things in mind as we move forward. So where do we stand with our investments in FASTR flu vaccines? As I said, we're probably going to make modest investments over the next year or two, helping to perhaps support some of the next generation technologies as they advance through early development. We have made an award recently, you've probably seen it, to Arcturus Therapeutics for implementation of their self-amplifying mRNA platform with flu. Additionally, in our pandemic vaccines and adjuvants branch, we're doing some work internally with one of our partners, Seqirus, looking at their self-amplifying mRNA platform as well. Typically, this early stage work is not something that BARDA gets into, so this is not something we'll likely invest heavily in, but I think helping to advance and seed the pipeline will pay benefits for us in the future. Another mechanism we're using to help advance this technology is through a collaboration we have now established with JPEO. I think many of you are aware that we collaborated with them in terms of the contracts that were awarded under the original Operation Warp Speed COVID effort. And we're now collaborating with them on improved flu vaccines as well. That collaboration is actually managed by JPO, and we are working with them now to make some initial awards, but you'll be seeing some more news about that in coming months. Okay. So the second area of interest, formulations for FASTR impact. The goal is, when looking at a vaccination campaign, not just to get vaccines in people's arms, but it's to elicit a protective response. So the faster we can elicit that response, obviously the more effective the vaccination campaign will be. One of the ways we think we can do that is by expediting distribution and administration, and potentially elicit a faster response by that mechanism. Again, this is a question or an approach that applies not just to flu vaccines, but to any other emerging infectious disease. And so we're actually collaborating internally within BARDA with our CBRN division and our DRIVE divisions to look at this, the potential for alternative routes of administration and their potential to benefit overall response capabilities. Our initial focus here is on microneedle patches. There was a source of SOT notice that was issued in September. That is not closed yet. I think it's closing in, actually maybe closing this week. So we're looking for potential partners who have capabilities to work in this area. We are interested in hearing from not just the companies who have the technology, microneedle patches, but also from companies that have influenza vaccine antigens that could be partnered with these patches. Marrying those two together is a key focus of our work moving forward in this area. Again, we don't look at this just in a vacuum. Although it may expedite delivery, we're hoping that perhaps patch delivery could improve performance of the vaccines, potentially enabling protection with one dose, for example. And so again, synergy is very important in the initiatives we're looking at going forward. And again, alignment with overall goals for emerging infectious disease preparedness continue to be relevant here. Yeah. So the third goal, more broadly effective vaccine designs or formulations. As I mentioned, the ultimate goal here would be something like a universal flu vaccine. Realistically, we don't see that as being a short-term goal, and we don't see the likelihood being high of programs ready for advanced development in that area. So we're focusing on more interim goals here. So one possibility would be formulations that elicit a protective response with one dose, not necessarily through an alternative delivery device, but through some sort of delayed release, for example. There could be a number of possibilities by which one dose could potentially result in protection rather than two. Another approach might be to look for a broadly priming vaccine that could be administered to people well in advance of a pandemic, so that we would only require then one dose of a specific pandemic virus vaccine to elicit full protection. We're also continue to be interested in antigen-sparing approaches, which we have been working on since 2006 as well. I think, as you all know, our current pandemic flu vaccine formulations all include adjuvants for antigen-sparing purposes, and we're not dissatisfied with the current adjuvants, but if there were adjuvants out there that would have either logistical or cost advantages or performance advantages, we would still be interested in hearing about those as well. And again, I would emphasize here the potential synergies between this area of interest and the others that we've talked about. And finally, the fourth area of interest, as I mentioned, this is a sort of corollary to the others. As we bring these new technologies on board and work with companies to implement them in our pandemic preparedness infrastructure, we will need to ensure that we have manufacturing capability available at sufficient levels to make it work during a pandemic outbreak. We've done this with our current vaccine formulations, and we will, of course, further down the road look to do that with these new technologies as well. So, for people out there who may see in those areas of interest potential to work with BARDA, I'll just quickly remind everyone that the Vaccines Research and Development Branch has an open, broad agency announcement with two areas of interest. And this is the way that you can approach us to actually put a proposal on paper and have us review it. Again, those of you who have looked at this know it's a two-stage process. Typically starting with a white paper that allows us to review and give you comments. And then if we look at your white paper favorably, then invite a full proposal, which we can then consider for funding. The areas of interest here have just recently been updated, but they have not substantially changed from the past few years. But I would remind everyone that while these were closed for some period of time, we were only accepting COVID submissions. All COVID submissions are now closed, and we are only accepting flu submissions. Having trouble getting into it. There it goes. All right. So this is a slide that you'll see probably in a number of presentations today. This just emphasizes the normal pathway that we see for companies to partner with BARDA in terms of where in the development cycle we would typically get involved. So you see on the left, we typically want to see preclinical data. We want to see some assessment of the production feasibility of a potential countermeasure, and we want to see plans for achieving that, both in terms of clinical and manufacturing efforts. At that point is when we would typically want to consider a proposal for BARDA funding, and then if that if that funding is awarded, then we would proceed to complete the development of the countermeasure through clinical, manufacturing, and licensure. So again, this is sort of standard BARDA practice and has been for the past 15 years. Okay, so with that, I'll turn the microphone over to Christine for the update from her branch.
Okay, thanks, Jonathan, very much. So my name is Christine Oshansky. I'm the chief of the Pandemic Vaccines and Adjuvants branch in the Influenza and Emerging Infectious Diseases Division at BARDA. And I wanted to give you all an update on our program entitled the Medical Countermeasures for Pandemic Influenza Response. Here we go. So our branch's goal is to advance the timely and accessible vaccination strategy to mitigate the next pandemic, or at least the next response. So on one hand, we look to accelerate the pandemic response timelines where we're able to. So we do gap analyses and try to close those gaps, whether they're small or large. As part of the program, we manage the U.S. National Pre-Pandemic Influenza Vaccine Stockpile program. And this stockpile it has bulk antigen and adjuvant, enough to vaccinate the CDC-defined Tier 1 population very early following a response. As part of this program, we also maintain a library of influenza virus vaccine seeds so that those are ready if needed. And most importantly, we have partnerships in place with the FDA-licensed influenza vaccine manufacturers, and that includes CSL Seqirus, GSK, and Sanofi. And in these partnerships, we're able to expedite procurement of enough vaccine for the U.S. population very soon following a pandemic declaration. And then on the other hand, we also want to make sure that the vaccine is accessible to all populations. So we work very hard to ensure that the vaccine implementation strategies that are in place remain nimble and flexible. We look to expand safety and immunogenicity data sets for influenza vaccines targeted to diverse subtypes. So we have a lot of data on H5 and H7, but not so much on H2 and H9 and some of the other subtypes. So we're looking to expand those data sets. And we also proactively ensure that the antigen that's in the stockpile remains safe and immunogenic because it is stored over time. Not sure where to point. Okay. So we've had quite a few successes since the advent of our program. So Jonathan mentioned that BARDA began in 2006, but our program actually began before BARDA was even BARDA. So we started in 2005. And since 2005, we've had contracts with the FDA-licensed influenza vaccine manufacturers, like I just mentioned, in place without interruption since the beginning of the program. And with them, I mentioned that we maintain a library of influenza virus vaccine seeds. We stockpile pre-pandemic vaccines as well as adjuvants. And this has all led to an antigen-sparing adjuvanted pandemic vaccine strategy that's already in place. We have key inter-USG agency partnerships, so we work very closely with CDC and FDA, as well as NIH, and through these partnerships and through BARDA's partnerships with the manufacturers, we were able to respond to the 2009 H1N1 pandemic and prepared to respond to the 2013 and 2017 H7 and 9 outbreaks several years ago. And we were able to use our program to award one of the first COVID vaccine contracts because our partnerships were already in place. We also work very closely with the Pharmaceutical Countermeasure Infrastructure Division. Okay, thanks. And either we have the partnerships directly with the manufacturers or we work with PCI, but we maintain pandemic readiness, including adjuvant vendor managed inventory and our year-round supply of eggs in case we need to ramp up production at any time. So we do have available medical countermeasures. So of course we have seasonal flu vaccines. We're hearing a lot about that in the news right now. We also have H5N1 licensed vaccines. So CSL Seqirus has their Audens. GSK has their H5N1 vaccine, also known as Cupan, and Sanofi also has an H5N1 vaccine. We also have licensed outpatient treatments, and that includes also Tamivir, Zanamivir, Baloxavir, and Peramivir. And I think Kim might touch on a little bit on this in her talk. But despite having all these available medical countermeasures that we already have, there do still remain gaps in our readiness. So, for example, safety and immunogenicity data for all the influenza viruses with pandemic potential, we don't currently have that. So if we do have that, we're able to reduce timelines because we're able to make a data-driven decisions when the time comes. And we're also looking to expand vaccine licensures. So H5 vaccines are already licensed, but we'd like to collect enough data to get additional influenza virus strains and subtypes into those licenses, continue our mix and match program where we're mixing and matching antigen and adjuvant, and then making sure that special populations are included in these data sets. And so that would include older adults and pediatric populations as appropriate. So I want to come back to this. I mentioned that one of our goals was to accelerate pandemic response timelines by ensuring that our implementation strategies, when we do go out to vaccinate, remain nimble and flexible. So I just want to highlight a couple of ongoing projects in our group. Jonathan's already touched on these a little bit because we work with their branch so closely. So we have the heterologous prime and boost vaccination strategy program, where we're looking to see how far we can cover antigenic space in terms of the immune response. So this really builds off of data out of the University of Cambridge through Dr. Derek Smith's lab. So BARDA sponsored a heterologous prime and boost clinical trial several years ago, where we were looking at heterologous H5 antigens and testing those. And we found that 2 doses of a homologous antigen given 21 days apart and then boosted about 4 months later with a heterologous H5 antigen really expanded the antibody response in those groups receiving those vaccines. So we're going to continue this program. CSL Seqirus is going to start a clinical study where they're looking also at 2 heterologous H5 antigens, and so that will be starting in 2023. And then we also want to move forward with this central antigen hypothesis. Jonathan talked a little bit about this too, where we think that a single antigen, whether it's protein or mRNA based, could induce broad immunity across H5 antigenic space when boosted with something maybe out of the stockpile. On the other hand, we are very interested to incorporate, where we're able to, next generation vaccine candidates. So our program is really focused on licensed manufacturing processes, but we want to use our program to incorporate where we're able to these next-gen vaccine candidates. And so one example of this is that CSL Seqirus is they're sponsoring a clinical trial where they're testing their H2 vaccine candidate. So on one hand, they're looking at the cell-based adjuvanted influenza vaccine, but we're also incorporating their next-gen self-amplifying mRNA platform into that study. That also begins in 2023. And then finally, we play a supportive role as well. So we are supporting the vaccine development branch and helping them to work with Vaxess in developing a skin patch design to self-deliver influenza vaccine. So I want to switch gears quite a bit, and I'd be remiss if I didn't talk about the 2022 highly pathogenic avian influenza, H5N1, that keeps popping up in the United States and elsewhere in North America. So the map on the left is a little outdated. It was back from August when these slides had to get submitted for clearance. But if you look at I have the website here. If you look at the website, the map looks a little bit worse than what I'm showing here. But just a little bit of background, highly pathogenic avian influenza, H5N1, of the Eurasian lineage was detected in wild birds in the U.S. earlier this year in 2022. And since then, it's caused outbreaks in commercial poultry facilities as well as backyard flocks. And I'm also reading of late wild bird markets in New York and in New Jersey. There was the first human infection with this H5N1 reported in the U.S. in April of 2022. That was in Colorado in a poultry worker. And likewise, sporadic detections keep popping up in non-human mammals. So that includes foxes, otters, and seals. And so the surveillance workers are really monitoring this very closely because this indicates a potential for widening of the host range. So it's being monitored. So during all of these surveillance activities, we were moving forward with our vaccine manufacturer partners. We've asked them to go ahead and please make seed lots to be prepared if we need to respond. And we've also partnered with CSL Seqirus and with GSK to clinically assess the safety and immunogenicity of the 2022 H5 virus vaccine candidate. And those studies also begin in 2023. Okay, so I'll step 2 steps back and just give you a program overview on this slide. So we work with CDC, the WHO collaborating centers, and other surveillance agencies to assess what's going on across the globe. Risk assessments are performed, and based on those risk assessments, the WHO collaborating centers or the central research laboratories, they begin making candidate vaccine viruses to put into the response arsenal, I guess if you want to call it that way, as well as potency reagents. As the risk is, as we monitor the risk, like I said before, we ask the vaccine manufacturers to go ahead and produce seeds. Sometimes they produce bulk antigen and sometimes final container if we're going into the clinic. And anything that's manufactured is put into stability programs. So we've had antigen in the stockpile since 2006, and so we have a nice data set, a long-term data set of antigen stability. We perform clinical trials, and then all the safety and immunogenicity data from these clinical trials, either BARDA sponsors these trials, the manufacturer sponsors them, or NIH sponsors them. And this data gets incorporated into pre-emergency use authorization packages that are already drafted, so these are periodically updated as the data becomes available. And so collectively, all this data and information, we hope that it leads to faster to first doses out the door, faster to 600 million doses. That's assuming that everybody in the United States needs 2 doses, and then of course faster to full immunization. So going forward, just over the next year or so, we're going to continue our risk-based monitoring for pandemic preparedness. We're supporting expansion of pandemic influenza vaccine licensures to get that additional data, those additional data sets to incorporate into these packages. We want to perform even more clinical trials and other data analyses on the existing samples that are available so that we can enrich the safety and immunogenicity data set. We'll continue to develop more candidate vaccine viruses and seed lots to promote pandemic preparedness. And of course, we provide support for and continue to collaborate with the other branches and other divisions in BARDA to support modernization of flu vaccines and decrease bottlenecks. And lastly, we want to use our program to provide opportunities to add improvements in technology. So Jonathan talked a lot about this already, but we can incorporate some of that into our program as well. So that includes any technological improvements, whether small or large, that can decrease response timelines, single-dose formulations, alternative potency assays, and even newly licensed influenza vaccines. So as new vaccines come forward, we would like to incorporate them into our program in some way. I think that is my last slide. Okay, I think I'll stop there and turn it over to Kim.
All right, so now we're really switching gears away from vaccines and into the therapeutics program. So my name is Kim Armstrong, and I am the branch chief of the therapeutics program within the Influenza and Emerging Infectious Diseases Division. And so, of course, our primary focus is, of course, pandemic preparedness. And we are incredibly lucky in the flu space to have a number of treatment options that Christina's already talked a little bit about, but we have drugs with 2 different mechanisms of action, as well as a number of licensed seasonal vaccines, which is really fantastic. But there are several gaps in the therapeutics program as well, and these gaps are really exacerbated in a pandemic situation. So the numbers are really quite large that you're seeing on the screen, but to put these into a little bit of perspective, during the COVID-19 pandemic so far, there have been 97 million COVID-19 infections in the United States and over 1 million deaths. And those have occurred over the course of the last 2.5 years. And what we're projecting here would occur over the approximately 12 to 18 months. So it is really something that we need to prepare for in advance to have our best response available. There it goes. So we have 3 main goals, and we really want to address the gaps that we I showed on the previous slide. And so number 1, we need to find a treatment for hospitalized influenza. So we have tried at BARDA through a number of different clinical studies to try to get antivirals approved for the treatment of hospitalized influenza, and all of these studies have failed. And so because of that, we want to refocus the program on host-targeted therapeutics that would be used as adjunctive therapy to antivirals and really have a broader indication, not just for hospitalized flu, but for acute respiratory distress syndrome, again, in the hopes of preparing us for the next respiratory viral pandemic. Another gap that we would like to address is pre-exposure prophylaxis. So we know from recognition of a pandemic to matched vaccine availability, there is a gap. And we believe a pre-exposure prophylactic that is broadly active against many different influenza strains could provide mitigation and fill this gap when in need. And then finally, we are always on the lookout for new antivirals for the treatment of outpatients with influenza infections. We really are interested in finding drugs with multiple mechanisms of action, so that really puts us in the best stance. No matter what pandemic virus comes forward, we will have many different treatment options, hopefully, to mitigate that. So our current portfolio is pretty large, but you can see it is COVID-19 heavy. The team is incredibly proud of the work we have done over the last 2 years to get these COVID-19 therapeutics across the finish line, but we're really excited to start focusing again on the influenza portfolio, and hopefully we can get that influenza portfolio to start looking much more like COVID-19. So I first want to tell you about our program with Genentech. So of course, Xofluza, or baloxavir marboxil, is one of the first new antivirals that was approved by FDA. And one of the best things about this is it is an endonuclease inhibitor. So this is the first new mechanism of action since the neuraminidase inhibitors were approved way back in the aughts. And so the other amazing thing about this drug is it is a single-dose antiviral. So from a pandemic perspective, this is amazing because you can watch your patient take that dose and you don't have to worry about patient compliance, which is incredibly ideal. And then most importantly, as you can see on the figure on the right, it is an incredibly potent antiviral. So what you're seeing is 24 hours after dosing with baloxavir marboxil, you can see a significantly reduced infectious viral load titer. And this is significantly different not only from placebo, but also oseltamivir treated patients. And this feeds really well into our pandemic strategy. So what you're seeing on the right is a hypothetical epidemiological curve of an influenza pandemic. So in red, you're seeing an unmitigated curve. And in yellow, you're seeing if we use antivirals early and often to perhaps prevent onward transmission, we could blunt that curve. And then you further combine that with non-pharmaceutical interventions and a vaccine, you can really blunt the impact of a pandemic. The biggest problem with this plan is that we have actually never proven that you can prevent onward transmission with any influenza antiviral. So this is why we're funding the Centerstone study in partnership with Genentech. And so in this study, we are treating the index patient who is influenza positive with either baloxavir or placebo, and then watching their household contacts to see if we can prevent that onward transmission with treatment. So we are excited to see this clinical trial is actually enrolling right now, and so we are hoping to see the results of this trial in the next couple of years. I'm not switching. So we are also always interested in additional antivirals to add into the portfolio. So like I said before, we're interested in new mechanisms of action, and we really are not considering neuraminidase inhibitors at this time. We want your drug to be commercially competitive, which means it has to be safe, just like oseltamivir is incredibly safe. And also the cost of your drug needs to be commercially competitive. You know, something that costs thousands of dollars is never going to be able to compete with generic oseltamivir. And then we also have some data requirements as well. So we want you to demonstrate superiority to oseltamivir in animal models before you come into BARDA for funding. And this is because we believe oseltamivir is the minimum bar to achieve clinical efficacy. And if you can't demonstrate superiority in animal models, you have a very low likelihood of success in phase 3 clinical trials. And of course, we're most interested in pandemic preparedness, so you also have to demonstrate that you have antiviral activity not only against seasonal influenza viruses, but also pandemic influenza viruses. So moving on to the pre-exposure prophylaxis program, we know that there is a 17 to 24-week gap from when we can recognize an influenza pandemic and when matched vaccine would be available. And so we think a pre-exposure prophylactic drug could fill this gap and particularly protect those who are most exposed, such as healthcare workers and first responders, but also protect the most vulnerable, such as the long-term care facility residents who really paid a heavy toll during the COVID-19 pandemic. In addition to pandemic preparedness, we really do think there's a commercial market for influenza prep. And so what you're seeing here is estimated vaccine effectiveness from the 2018 and 2019 influenza season. And you can see in those age over 65 who are highly vaccinated at around 82%, there was an estimated only a 12% adjusted vaccine effectiveness, showing that this is a highly motivated group to receive prevention, but also needs additional help in preventing influenza disease. So I'm incredibly excited to tell you that we have our first drug in the PrEP program in the influenza division, and that is in partnership with Vir to develop a VIR-2482, which is a half-life extended monoclonal antibody that targets a conserved region within the hemagglutinin stalk region of influenza viruses. So this is specific for influenza A and is active not only against seasonal but also pandemic strains including H5N1 and H7N9. It also has an intramuscular route of administration, which is really fantastic for pandemic operations and ease of dosing individuals in mass dispensing situations. And we're really excited that they are also enrolling in a clinical trial this Northern Hemisphere season as a proof of concept in their phase 2 clinical study. But of course, we're looking to expand this portfolio, and we are looking for drugs that where one dose of the drug will provide at least 6 months of protection. This will provide protection for the entire influenza season, and we're hoping it could be given in conjunction with the influenza vaccine. We want a drug that will prevent against both influenza A and B because we think that will be the most commercially competitive. But of course, VIR-2482 is only active against influenza A, and that does meet our minimum bar for meeting our needs. And again, we're targeting an efficacy where there would be a 70% relative risk reduction of infection in those who do not respond well to the influenza vaccine, such as in the elderly or the immune-compromised populations. But in addition to this study, which would likely be your pivotal trial and gain licensure, we're also going to be looking for data supporting use in healthier populations for pandemic preparedness. We want your drug to be commercially competitive, not that you have to be the same price as a vaccine, because that's pretty high bar to meet in this space. But something that costs tens of thousands of dollars is not going to be amenable for yearly dosing in this market. And in addition to the long-acting antibodies, we do think a controlled release drug may be a similar option in this space, but we're always looking for new ideas. And if you want more information on BARDA programs for pre-exposure prophylaxis, please check out the breakout session that is tomorrow at 11 a.m. And then finally, I want to talk a little bit about our hospitalized influenza gap and how we're going about that. So we know that 10% of ICU patients at any given time are suffering from ARDS, and those patients have about a 40% in-hospital mortality rate. But to date, there are no FDA-approved drugs or treatments for ARDS, and so we're really looking to fill this unmet medical need with host-targeted therapeutics that would again be used in combination with standard of care, such as antivirals or antibiotics. And we're really excited to be doing this in the influenza program because this will not only fill the needs, for influenza, but perhaps for the next respiratory pandemic event, but also other programs within BARDA who also have lung complications. So using host-targeted therapeutics in a complicated disease really does come with a number of challenges. And this is demonstrated by industry's history of failure in both sepsis and ARDS, right? And so this is really hard to overcome as a lot of the major players are just not interested due to this long history of failure of these clinical trials. And then another issue is really defining what patient population will benefit from your drug and are there triggers for treatment. So you can think about in the diseased lungs, there's many different things going on. And many patients are coming in and they're in this hyperinflammatory state. So if you treat them with corticosteroids, you could really see some benefit as we saw in COVID-19. However, other people are coming in and they're in a hypoinflammatory state, and corticosteroid treatment can actually cause harm, as has been demonstrated in multiple influenza clinical studies. So this is a very complicated space to try and think about how to develop a drug. In order to get a handle on this, we are thinking about starting a phase 2 screening trial for treatments of ARDS with 3 main goals. Goal number 1, of course, is we want to test candidate therapeutics in this space and really find some data that provides us a reason to move forward into phase 3. And then our second goal is to generate a lot of biomarker data so we can really hone in on what is a specific population that your drug can benefit, so that you can design your phase 3 appropriately in a way that you can are most likely to achieve success. And then finally, we really want to learn more about the disease and really define clinical endpoints and find ways to get a drug across the finish line. So we did have an open RFI, which is now closed, and I'm hoping colleagues can put the link to the RFI in the chat, because our protocol synopsis is still there, so you can take a look about what we're thinking about here. In addition to the phase 2 clinical trial, we're also interested in just funding drugs in the advanced development for ARDS. So of course, we're looking for pathogen agnostic treatments. So we will not consider antivirals or antibiotics in this topic area. However, because we're using influenza funds, you really must demonstrate relevance in an influenza animal model or in clinical data showing that your drug can benefit an influenza-infected patient. In addition, we want to understand how you propose to get your drug across the finish line, and we're really hoping you're not trying to treat every ARDS patient that walks in the door, because we think this is one of the big reasons why all the previous studies have failed in the past. So really coming in with a targeted plan of who you intend to treat is more likely to meet with success from our reviewers. Now, we're taking the biomarker approach, but it could be a different approach. You could use comorbidities, to really create a more homogeneous population. And then finally, you must have an open IND for the treatment or prevention of ARDS with the US FDA. Anything specific to influenza will not be accepted in this topic area. And if you're interested in learning more about host-targeted therapeutics and what BARDA is doing in that space, please check out the breakout session that is occurring tomorrow at 3:15. So I have a number of requirements for BARDA funding. I don't want to repeat what I've already said, but number 1, before you come in, you must have a phase 1 clinical trial complete and data in hand before you submit your proposal. We really want drugs that have already demonstrated safety before they come to us for funding. And if you are missing any of the data that is listed on this slide or that I've talked about previously, please reach out to Nita Basu over at NIAID. She has a portfolio of preclinical drugs in the influenza space, and she's always looking for new candidates to add to her portfolio. So I really want to thank you all for your attention during this session and in this really warm room and staying awake. It's it's been really great, but we're happy to take your questions.
Hopefully everyone can hear me. I can hear myself. All right, so the first question that we have this afternoon is for Jonathan. Going forward, will BARDA consider any novel flu vaccine technologies that are not RNA-based?
Sure. So I emphasize RNA platforms simply based on the COVID experience that they've shown under fire that they can actually do the job. But we're not ruling out other platforms that could be equal or better. Obviously, we looked at viral vector vaccines in the COVID response as well. They encountered some issues, so certainly if you want to bring something like that forward, you'd have to address the concerns that have been raised, or any other platform as well. So yes, we're open to them, but right now mRNA is the benchmark.
Thank you. I just never am tall enough. Thanks. Next question is for Kim. In reference to the need for broad antiviral neutralization to demonstrate preclinical antiviral activity against pandemic avian strains, how many strains need to be tested?
So prefer as many as you can is the kind of preferred answer, but at least one from group 1 and one from group 2 is absolutely mandatory.
Great. All right, we'll pitch one over to Christine. Can you please describe the difference between the pandemic influenza vaccine stockpile and the strategic national stockpile?
Sure. So the Strategic National Stockpile, or the SNS, it is an important tool for our nation. It includes supplies, drugs, and devices needed for life-saving care, but they don't include non-finished goods. And so that's why we have the National Pre-Pandemic Influenza Vaccine Stockpile. So in this stockpile, it's bulk antigen. And that way we know in a response we can finish it at a certain potency because we're testing it at that moment in time.
Thank you. New question for Kim. Will BARDA consider support for new antivirals with novel mechanisms of actions which only have animal tox safety data prior to phase 1 results?
So the therapeutics program is really limited. We really are in the advanced research and development space, and so you must have that phase 1 data in hand and an open IND with the US FDA before coming into the BARDA BAA. However, there are a number of other places that you can go to help to get some resources for those, which number one was NIAID, and I gave you the contact information before, but also DRIVE may have some available topic areas that can also help get you across that finish line to that phase 1 data.
Great. Question for Jonathan. Can early-stage vaccine development programs be supported by BARDA under Area of Interest 8.2 since there is no TRL restriction?
So no, that has caused some confusion in the past. There is no TRL limit on Area of Interest 8.2 because it's not intended to support full vaccine development. So there is no product development, there are no TRLs that are relevant. What we're looking for under 8.2 are refinements, enhancements that could be applied, for example, to existing vaccines that are already licensed that would improve manufacturing efficiency or productivity or other aspects. But yes, if you have an early stage vaccine development program, it would not be accepted under 8.2.
Thank you. And a question for Christine. Potent adjuvants induce cross-strain protection in influenza and several other viral infections. Is there a plan to stockpile adjuvants?
Yeah, so we have adjuvant in the stockpile, of course. We're interested in new adjuvants, so I don't know if, Jonathan, you want to comment any more, but adjuvant can be part of one of the areas of interest in the BAA. But we can certainly talk to you about incorporating that into some of our clinical trials. Sustainability is the key here. So we don't want to stockpile something that's not sustainable, of course, and so it should be part of a licensed influenza vaccine, preferably.
Yeah, so as I mentioned, adjuvants are still of interest to us, but they would have to be some improvement over the existing vaccines. The adjuvants that go into our vaccines. So you'd have to identify that and make a strong case for why we would want to invest further in adjuvants when we already have ones that work. But if you do have something along those lines, we're certainly willing to consider it.
Great, thank you. For Kim, for the ARDS therapeutic, does the ARDS origin have to be from influenza or could it be from other origin? And does the phase 1 data require phase 1 data with interim report is okay for submission, if you could confirm.
Sure. So we do not want your phase 2 and phase 3 trials to be influenza specific. So we want the influenza data to justify funding, but we want a very broad indication of acute respiratory distress syndrome. So yes, we do not want influenza specific patients. We will accept an interim report from a phase 1 clinical study as long as it covers the dose and duration of treatment that you're proposing to move forward with in phase 2. So we have that data in hand to make an informed decision on how your program can progress.
Thank you. There are some questions with regard to how industry partners can reach out to discuss the products that they're developing. Do you have a recommendation for how they could reach out to you for antiviral development?
So TechWatch is actually the best way to reach not only BARDA, but a broad U.S. government audience to really talk about your program. And that is by far the best way to reach out to us. I did provide our email addresses, so you are welcome to email me and the team. But I will say the TechWatch team is very responsive and is the best way to reach to us.
Thank you. This one may also be for therapeutics, though I just lost it, and I apologize for that. We'll have to come back to that one. In the meantime, Jonathan, someone is working on a universal flu vaccine design, and they're wondering if this would fall under BARDA's areas of interest at this time.
We would love to have a universal vaccine program that is ready for advanced development. That's the ultimate goal, is to have a universal flu vaccine. Our assessment of the landscape is that that's unlikely in the near term, but I'm not foreclosing the possibility that if someone can surprise us with a technology that appears likely to succeed and is at an appropriate stage of development, we would certainly consider it. Certainly another aspect of all the work that's going on as far as broadening the protective scope of flu vaccines, so working toward a universal flu vaccine, nothing is really crystallized in terms of what looks like a highly likely path to success. There's still many different avenues that are being investigated. So again, this contributes to our thinking that this probably is a longer term consideration and not something in the short term. But again, we're not foreclosing it, but you'd have to meet the criteria.
Great, thank you. All right, 2 quick questions for Kim. First, is it possible to use combination of drugs with different mechanisms of action together?
So for ARDS, absolutely. I think it will make some of our regulatory colleagues feel very nervous when we think about combination rule, but this might be a viable path forward. So it's absolutely allowed.
Perfect. And in the last few seconds, why have we limited the PrEP effort to products which require only 1 dose with a 6-month duration?
Yes, so our minimum requirement is actually 1 month, a duration of protection for 1 month, but we want that 6 months, and that is really just to coincide with the influenza season. The influenza season is 6 months long, and it ideally, like I said, be given at the same time as an influenza vaccine, and really used every year. So when a pandemic comes around, doctors know how to use it and are really comfortable with its use.
Fantastic. Thank you so much.
Thank you.